Good morning to everyone and welcome to my poster, which is about a transcriptome-wide prediction and interpretation of translation states using sequence-to-sequence learning. In recent years, our ability to measure gene expression mainly relied on RNA-seq. Despite this technique being really good, it fails to monitor the translational output of mRNAs, so we don't know if an mRNA is actually translated into a protein or not. A technique that we can use to understand this is ribosome profiling, which maps the position of the translating ribosome at the single nucleotide level. And this allows us to understand and comprehend complex translational dynamics and some events like ribosome scanning, posing, or colliding ribosomes. What we plan to do is to use natural language processing methods and convolutional neural networks to understand which are the relationships between some RNA features, like the RNA sequence or the RNA structure, and the riboseq profiles that we get from the experiments. Before doing this, we have decided to use in silico experiments that allow us to better understand and interpret what the models are doing. And we plan to do this on tasks that are progressively more complex. So we started, for example, from plot A here with some peaks that are shifted with respect to some specific sequence patterns that we have decided, and more complex signals that resemble a riboseq experiments where we have, for example, upstream morph, coding sequences, and other different type of signals. And we want to do this using different types of architectures such CNN and gated recurrent units from the recurrent neural networks. Here we can see the performances where the gated recurrent units tend to perform better than the CNN or the mixed network. And here we can see that from the dataset where we have peaks, we can see here an example. The CNN tend to be more noisy with respect to a gated recurrent unit. And then we have an attention map. The attention map is very crucial to us because that's what allows us to interpret what the network is doing and why he is predicting some things over others. But we can get this only from the gated recurrent units. Instead, here from the convolutional neural networks, we have decided to try in silico mutagenesis, where we progressively mutate every nucleotide in the sequence and compare the output of the network on the mutated sequences with the original sequence to understand which part of the sequence are actually important for the model. On the other side, from the riboseq-like signal, we can see here an example and the prediction. So the model, which in this case is a gated recurrent unit, is able to identify almost everything that we put inside of our in silico experiment. So, for example, the trinucleotide periodicity, which is characteristic of the riboseq, but also the upstream morphs, some particular type of noise where we have provenance, there is a higher signal, a lower signal for some nucleotides when there is the depletion, which is on a certain codon. And here we can see how the attention is on average around some patterns, which are, for example, the starting codon, which has a very strong attention where the codon is, and a periodic signal after the codon, which is not present before the codon. And the same is true for the stop codon, which has some peculiar shape after the stop codon, and the proline, which again has a pattern. And what we would like to do is to better understand what the model is looking at when it's doing its prediction. And so we want to extract the rules that we gave to our in silico experiments from the attention matrices, and this is a tentative example where we have the attention matrices that has been clustered for each nucleotide, and in this way, from the UMAP, we can see that we're able to distinguish the attention on the 3' UTRs, on the main ORF, and on the 5' UTR. However, we're not as good to understand the differences between the attention on the starting codon and the stop codon, which are a bit mixed on the UMAP. Our plan in the future is to have, let's say, a different protocol. So we want to actually have a protocol in which we have like where we have more than one signal. The signal that is coming from the riboseq data has fragments that are 30 nucleotides long, but with a modified version of the protocol we're able to get different fragment sites, which represents colliding ribosome, for example, or rotated ribosome that are starting the elongation phase. And we want to have like a problem in which we have multiple signals, and so we get more data, and maybe we will be able to elucidate better the molecular mechanism behind the translation cycle. And to conclude, I would like to say that we would like to use also transformers in the future, beside recurrent triangle networks and CNN, because we know the limitations of these networks. The CNN, in our case, it's not easy to interpret because we don't have an attention map, and the gated recurrence units will become increasingly more complicated to use if the sequences gets longer. And thank you for your attention, and goodbye.